This is Chapter 7, the cellular basis of inheritance. So in this chapter, we're going to be looking at sexual reproduction. And so this will be how we get some genetic variation. Here in this picture on the left, you've got three different examples of complex cellular organisms. So sexual reproduction is what has allowed us to evolve more and be able to have more complex multicellular organisms. So with sexual reproduction, you have the production of haploid cells by parents, and those two cells are going to fuse to form a new unique diploid cell. It provides much more genetic variation than mitosis alone. So this is called meiosis, and it's division of the chromosomes among the gametes. So mitosis was division of the nucleus. Meiosis will be division of the chromosomes. So different organisms do have different sexual life cycles. A sexual life cycle is when fertilization and meiosis alternate. Meiosis is going to reduce the gametes' chromosome number by half, and then fertilization will join the gametes and restore the diploid condition. So organisms that are diploid dominant are going to be multicellular in their diploid stage. It's their obvious life stage. This is going to include humans and animals. So you spend the majority of your life as a diploid adult human. The haploid dominant would be multicellular haploid stages, and that will be their obvious life stage. This would be fungi and some algae. So this would be like multicellular sperm or multicellular eggs. With alternation of generations, you have both haploid and diploid stages that are apparent. And this will include the plants and some algae. So a germ cell is going to be the special line of cells that will produce gametes. The gametophytes are going to be the haploid multicellular plants. These will produce gametes. And the organisms that produce the gametes are already haploid, so no meiosis is needed. When you have sporophytes, the zygote undergoes multiple mitoses to form multicellular diploid plants. So you'll have special cells that will undergo meiosis and produce haploid spores. Sometimes these cycles are easy to compare with a visual rather than just with words. So picture A on top, you've got the animals where they're going to do sexual reproduction as adults and form haploid gametes, your sperm and your egg, that will unite to form your diploid germ cells. With the fungi, such as black bread mold, these are going to have haploid dominant life cycles. So they will spend the majority of their life as haploid cells. And then with plants, you can see that this division line is right in the middle. They'll alternate between multicellular haploid organisms and multicellular diploid organisms. So fertilization is the union of two cells from individual organisms. Remember, your somatic cells are your non-reproductive cells of the multicellular organism. That's everything except your sperm and egg. Meiosis has two main phases. You've got meiosis 1 and meiosis 2. In meiosis 1, it's the first meiotic division. It's going to reduce the number of chromosome sets from 2 to 1, and this will make recombinant chromosomes. Meiosis 1 can be subdivided to have prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis 2, your second round meiotic division will take place similar to mitosis. It's your equatorial division. And in here you can have prophase 2, metaphase 2, anaphase 2, and telophase 2. This is showing crossing over. You have a blue chromosome and a red chromosome here. And what happens with crossing over is you have a piece of the chromosome that will switch with its homologous chromosome. And so what you end up with is two new chromosomes here that are going to be recombinant, along with the non-recombinant chromosomes on the other half. This is going to give more variety to the offspring. So instead of inheriting everything that is solely from one of your parents, when you pass down your chromosomes, your two pairs will be a mix of what you inherited from your two parents. So in interphase, we still have the same three parts, G1, S, and G2 phase. G1 will be focused on cell growth. S phase is when the chromosome is replicated or does chromosome synthesis, and G2 is your final preparations for meiosis. So what's going to happen is you're going to have random assortment that is independent for each chromosome at metaphase 1. So here at the top you've got metaphase 1. You can see you've got a red and a blue for chromosome 1, a red and a blue for chromosome 2. So one possibility is that both of them end up going this direction. It's a 50-50 chance that one of these cells will get the blue one, a 50-50 chance one of them will get the red one. When you get to the next chromosome, it's a 50-50 chance there as well that you'll get the blue one here or the red one here. These will undergo meiosis too, and then you will have just half of each chromosome being split into the germ cells. So what you can see is this leads from one germ cell to 4 haploid cells, where with mitosis you had one cell that led to 2 haploid cells. So the other option is you can have, with the 50-50 chance, one is going to get a blue and a red, the other one will get the red and the blue. So here you've got 4 different possibilities that will occur between these two examples. So with more chromosomes, the number of possible arrangements will increase dramatically. So meiosis 1 is your reductional division. Here you've got synapsis, which is the tight pairing of the homologous chromosomes. The genes are going to precisely align so they can pair in the same spot. You'll have crossing over where you will exchange bits of the chromosome segments between the non-sister homologs, and this will create greater genetic variation. Your recombinant sister chromatid has a new and unique assortment of the maternal and paternal genes. So along with having crossing over, making your recombinant chromosomes, and then having your independent assortment where you'll have a unique assortment of maternal and paternal chromosomes end up in the gametes, this creates a lot of variation. So in humans we have 23 chromosomes with 2 possible alignments. So we have 2 to the 23rd possible combinations, or over 8 million possible combinations. So if you were to look and you said, you know what, I really like the way my first child turned out. I want to produce the same exact chromosome set in another egg to produce another one of these children. It would be 1 in 8 million chances that you would have the same sets of chromosomes assort. It's even greater variation when you would take into account crossing over. The variation gets even greater when you consider that that is also going to be paired up with somebody else's germ cell, the sperm or the egg. So in meiosis 1, you're going to have 4 subphases to it. Prophase 1, where you're going to have synapsis and crossing over. Metaphase 1, you'll have the independent assortment. They're still going to line up along the metaphase plate. In anaphase 1, the homologous chromosomes separate, and the sister chromosomes are going to remain attached at this point. During telophase 1, the chromosomes are going to arrive at opposite poles. They will decondense, and you'll make a new nuclear envelope. And you'll have cytokinesis that will divide the cytoplasm, either creating a cleavage furrow or a cell plate. So here it's showing in meiosis 1 that you've got your microtubules that will attach to the fused kinetochores of the homologous chromosomes. In anaphase 1, the homologous chromosomes are going to separate. In prophase 2, the microtubules attach to the individual kinetochores, and you'll have the sister chromatids separate. So meiosis 1, homologous chromosomes separate. In meiosis 2, sister chromatids separate. So mitosis 2, your equatorial division. Your sister chromatids are going to form the haploid cells and split into 4 haploid cells. In prophase 2, the chromosomes are going to condense the nuclear envelope fragments. In prometaphase 2, your nuclear envelope disappears and your mitotic spindle is formed. Metaphase 2, the sister chromatids align. Anaphase 2, the sister chromatids separate. In telophase 2, the chromosomes are at opposite poles and decondense, and you have a new nuclear envelope form. And finally, it ends with cytokinesis, where the cell separates into 4 genetically unique haploid cells. So your key features that are different here is that it's the sister chromatids that are going to align and separate in meiosis 2, where it's the homologous chromosomes that align and separate in meiosis 1. So this diagram here is going to show you both meiosis and mitosis so that you can see a comparison. You can see both are going to have those 4 stages that get repeated. With meiosis, you end up with 4 haploid cells. With mitosis, you end up with 2 diploid cells. Here, your offspring are genetically identical to the parent cells. Here, your offspring are unique in meiosis. So both meiosis and mitosis will divide the nucleus. Mitosis is just a single division where you get a new genetically identical individual that is the same as the original, has the same number of sets of chromosomes. In meiosis, you have 2 nuclear divisions. It's going to form 4 new nuclei or cells. They are never genetically identical, and they have half the number of chromosome sets. So a karyotype can be used to identify errors in meiosis. When they're aligned in the chromosome, they pair with the same length, same banding pattern, and same centrosome position. With a karyotype, you can compare the chromosomes and make sure that you actually have 2 homologous chromosomes for each of the chromosomes and detect chromosomal abnormalities. You'll actually have a lab activity where you'll get to practice doing this. So here's showing a karyogram of the chromosomes for a human immune cell during mitosis. So you can see same staining pattern, same length, going all the way through. There are 2 X chromosomes which make this one female. So we can end up with disorders in chromosome numbers. With the translocation, you have the segment of one chromosome break off and it will attach to another chromosome. When you have a non-disjunction, you have pairs of homologous chromosomes or sister chromatids that fail to separate. You see increased incidence of this with increasing age of the parent, particularly with increasing maternal age, although it does increase slightly with paternal age as well. A euploid is an individual with an appropriate number of chromosomes for their species. An aneuploid is an individual with an error in the chromosome number. When you have monosomy, you have a loss of a chromosome. When you have trisomy, it's a gain of an extra chromosome. A common example in humans is trisomy 21 or Down syndrome. Many of these monosomies and trisomies do not actually end up as viable organisms in humans. We're pretty sensitive to chromosome changes. So this is showing an example of a non-disjunction. So each gamete should have 1 copy of the chromosome, except you ended up with 2 of them going over here. So over here, these are only going to have a single one. Over here, they have 3. So each of these cells are going to have 3. They are considered n plus 1. Here, they're missing 1, so they're considered n minus 1. If you have the non-disjunction occur a little bit later, so here it went through meiosis 1 and everything was okay. If the non-disjunction occurs during meiosis 2, you have 1 over here, you have 2 and 1 on this side. Over here, everything's normal. So what you end up with is two normal gametes, but then you're going to end up with a gamete that is an n-1, and you'll end up with a gamete that's an n+1. One has an extra, one is missing one. So here you can see the incidence of trisomy 21 increasing with advancing maternal age. You actually see an increase in any chromosomal disorders with advancing maternal age, not just trisomy 21. Some other chromosomal disorders that can happen are with the X or Y chromosome. With X inactivation, this is going to occur in females that have two X chromosomes. One on each cell condenses to form a bar body, and the genes are not expressed. So this X inactivation is a normal thing that will happen in each of the cells that has an X chromosome. So you end up with only one of the X chromosomes being expressed. This allows the X chromosome anomalies to be milder, and the person can actually have a mosaic pattern where some of their cells are expressing the anomaly and some are not. So there's a 50-50 chance of which X chromosome is going to become inactivated in each cell. With the triple X, this is a female that has acquired an extra X chromosome. She will develop as a female, but will have developmental delays and reduced fertility. With Klinefelter syndrome, this is an XXY individual. They will be a male with small testes and large breasts and decreased body hair. With Turner syndrome, this is the only viable human monosomy. You have just one X chromosome. There's no second X chromosome, there's no Y chromosome. So these will be a female. She tends to be short, sterile, and having hearing and cardiac problems. So polyploidy is any individual with more than the correct number of chromosomes. So the embryonic inactivation of one of the two different X chromosomes is what will give rise to this tortoiseshell phenotype in cats. So that appearance comes from it being a random 50-50 chance of which X is going to become the bar body in each of the cells. We can have some chromosomal structural rearrangements. These can include partial duplications, deletions, inversions, and translocations. One example is Reproduction. There's a partial deletion of chromosome 5. The infants have a high-pitched cry. They say it sounds like a cat in distress. With a chromosome inversion, it will detach and rotate 100 degrees and then reinsert itself as part of the chromosome. With a translocation, it can be benign or it can be associated with several cancers and schizophrenia. So there are some types of leukemia that are associated with translocations. So this is an individual here with Reproduction shown at various ages 2, 4, 9, and 12. So here this is showing what happens with an inversion. You take this piece out, you flip it around, and stick it back in backwards so it's in the reverse orientation. With reciprocal translocation, this is between two non-homologous chromosomes, and it doesn't cause any genetic information to be lost or duplicated. So here you have a piece of this chromosome and a piece of this chromosome that are going to trade places.